Assalamu alaikum dear students. Hope all of you are fine by the grace of Almighty Allah. Welcome to NIEVS Homeschool. I am Naima Akhter Tesha, Assistant Teacher Science, Morning Shift of National Ideal English Version School. Today I am going to take class 8 science class based on chapter 2. Let's start our class. Dear students, is our lecture number 7 and today I will provide you is the video number 5 and today our topic is chapter 2 reading and it will be part 2. Let's see. In your previous class we have discussed about mitosis, meiosis, amitosis. Today I will discuss the steps of mitosis. Mitosis is one kind of cell division. Normally it's occur in somatic cell and this cell division is occur in two parts. At first nuclear division occur, then the cytoplasm division occur. The process of mitosis takes place in two phases. The nucleus divides in first phase and cytoplasm in second phase. The division of nucleus is known as cytokinesis, karyokinesis. The division of nucleus is known as karyokinesis. As nucleus carry the chromosome and division of nucleus means the division of chromosome, that's why it is karyokinesis, and the division of cytoplasm is cytokinesis. And in mitosis at first karyokinesis occur, then cytokinesis occur. Mitosis is a continuous process in which karyokinesis occurs first and the cytokinesis takes place and then cytokinesis takes place. Before starting the karyokinesis and cytokinesis, the nucleus of the dividing cell need to take some preparation. This intermediate stage between the two consecutive cell divisions is known as interphase or resting stage. Okay, that means mainly interphase or resting stage in cell division 90 to 95% time will cost in preparational stage and only 5% time cost in main cell division. At first a cell take preparation, that means which things are need for any cell division. At first the cell arrange them, then the cell division start. Now there are 5 phases of mitosis cell division: prophase, prometaphase, metaphase, anaphase and telophase. In here at a time the plant cell division and the animal cell division are given and they are similar, only we know that in animal cell centriole present near the nucleus, that's why that centriole will present here and it will go in next cell. Now let's see. In prophase, it is the longest phase in this mitosis cell division. At first the cell division start from mother cell. We know that cell division start from mother cell and here normally in the cell cytoplasmic organelle will present, but that division occur in later. At first we will discuss about nuclear division. Inside the nucleus, what you have learned the structure of nucleus in your previous class and also class 6 also. Nucleus contains nucleolus, nuclear membrane, nucleoplasm and chromatid. Here that means here at first the chromatin, here mainly the chromatin division we will discuss here, mainly the chromatin division. You can see the nuclear membrane and the chromatin, and in the prophase stage at first dehydration start. In prophase stage dehydration start and that's why from chromatin water will remove and gradually it become appear or it become clear, and then this chromatin will known as chromosome. If I draw the picture of prophase first, This is a cell. In the at the very beginning of the prophase, you can see like that. Here the nucleolus, and one part of a chromatin will attach to it nucleolus and other part remain opened, and the structure will like that, and this structure will known as chromatin. And when the dehydration start, you can see the main structure of chromosome. It is the second after starting the prophase. At first the mother cell will like that. When the dehydration start, gradually you can separate the chromosome or see the chromosome. Here it is nucleolus and chromosome become like that. Chromosome contains centromere, and here this portion are arm, the two portion. And when you draw the picture, at first how many chromosome you will take, you have to continue that chromosome number till the ending. Here I will take here 2 chromosome. Look here. At the first it is the mother cell and prophase start here, and dehydration start at the beginning, and after dehydration when it start this chromatin, you can easily see the shape. Here the centromere and 2 arm in both sides. You are seeing that. Okay, I am drawing that portion, and gradually at the ending of the prophase, here you can see the nuclear membrane is clear and nucleolus also clear, and chromosome are single. Now at the ending of the prophase, the chromosome will divide longitudinally. That means, suppose I consider this as a chromosome and it will divide longitudinally. Centriole is remain here, look here, one arm of a chromosome divide into 2, and this portion is divided into another 2, and it is united through centromere. And these 2 if is known, and these two and these two, if I consider, it is mainly 1 and it is 1, and this and this is known as sister chromatid. At the end of the prophase, chromosome will divide longitudinally and produce sister chromatid. This and these are sister chromatid, and they are joined in the centromere. Now at the end, here I have shown the main structure. At the end, the nuclear membrane start disappearing. Here, and nucleolus also start disappearing, but it will present up to pro-metaphase. Here at the ending it start disappearing, and chromosome are divided longitudinally into 2 chromatid. Here it is like that, here it is like that. Okay, now our prophase is finished. We will start pro-metaphase. Let's see the characteristic of prophase. The nucleus becomes bigger in size due to continuous dehydration. Nuclear chromatin condense to form visible thread-like structure known as chromosome. Then each chromosome divides longitudinally into 2 chromatid. The sister chromatids are joined together at their centromere. I have already discussed this portion. Now we will start pro-metaphase. from this we will go to pro-metaphase. That means this picture will be the first step of pro-metaphase. So at the beginning of pro-metaphase, what we can tell, nuclear membrane and nucleolus almost disappear, and new structure will develop. If we consider new structure will develop, and that is known as spindle apparatus. Spindle apparatus. Nuclear membrane and nucleolus will almost disappear, and new structure will appear, that is spindle apparatus. Spindle apparatus is like that. From one end up to another end it will spread. Spindle apparatus. Here the nuclear lus will present, but almost in disappear condition. Here we use 2 chromosomes, so I will show you here 2 chromosomes. Already they are divided into chromatid. One chromosome is here and another will be here. Spindle apparatus will produce, and this is and this ray are known will be known as spindle fiber. And here one chromosome is attached with one spindle fiber, here another, using the centromere, and the middle portion, and this portion, that means in the middle portion of this cell is known as equator, equatorial region or equator. Now let's see the feature of pro-metaphase. It is the very short phase. Nuclear membrane and nucleolus almost disappears. At the end of prophase, spindle apparatus is formed by the accumulation of some protein fiber in the cell. That means this spindle fiber is produced accumulating the protein of a cell. The middle area of 2 pole of spindle apparatus is known as equator. This one and this one are 2 pole, and middle area, that means this portion will be known as equator or equatorial region. This portion and this portion of this cell now known as pole, and the middle one is known as equator. The fibers of the spindle spread from one pole to another. Look here, from one pole the fibers are spread from another pole up to another pole. These are known as spindle fiber. In animal cells, in addition to formation of spindle apparatus, centriole divides earlier and reaches the 2 poles from where aster fiber radiate. In animal cell, in that portion and that portion, the centriole will go, and from that, look here, just like here, centriole will go that portion and that portion, and they will emit the ray. Now come to after that, after the formation of spindle apparatus and disappearing of nuclear membrane and nucleolus, it will go to next phase, metaphase. Look here, the picture of metaphase. So in the metaphase, if I draw the metaphase, only the spindle apparatus will present. No nuclear membrane and nucleolus you can find here. Look, it is one of the important phase. Look here, spindle apparatus will present here. This portion, that means this 2 side will be known as pole and this portion will be known as equator, and in metaphase, dehydration reached in final stage and chromosome become shortest, quite thick and clear. That means in metaphase stage, chromosome are the smallest among these 5 phase. Chromosome are the smallest in metaphase stage, and they become more prominent, and they distribute in the equatorial region. They distribute in the equatorial region, okay, here and here. They distribute in the equatorial region. Let's see the characteristic. All the chromosomes come and located at the equator of spindle apparatus and get attached to the centromere by spindle fiber. Using the centromere, the chromosome are attached with spindle fiber. At this stage, the chromosome are found shortest, quite thick and clear. You can see here the structure. Then anaphase stage will start, and in anaphase stage already you can see in the prophase the chromosome are divided into sister chromatid, but they are joined using the centromere. In anaphase stage, the division of centromere occur and chromatid becomes sister chromatid becomes separated, and they transfer into sister chromosome or daughter chromosome or daughter chromatid. We can say them daughter chromatid. Look here. It is spindle fiber, and total structure will be known as spindle apparatus. And already the chromosome are divided. And they will like that. They are daughter chromatid. This, look here, there are two sides. It will divide longitudinally and turns to daughter chromatid. Now they will start to go. If they separate, suppose this, if I cut this, if I cut this like that, then two parts will form, and they will attach to it spindle apparatus, okay. Then, okay, no, it is not like that. They will attached with spindle apparatus, and they will start to go in two different pole. They will start to go to different pole, and for this kind of movement, centromere remain in front side and their arm remain in back side. Here look, at first two sister chromatid are separated and start going to different pole, and their centromere remain in the front side and in this movement, and their arm remain in the back side like that. And when they reach in two different pole, then the anaphase stage will stop and it will goes to telophase stage. And depending on the position of centromere, in that stage, in anaphase stage, we can divide the chromosome into 4 kind. If the centromere is remain at the middle, then when it start going, it will turn to this shape, that means V. If this centromere is present in like that, when it start going, it will turn to this shape, that means J like. It is V like. If the centromere is remain in like that position, it will, that means I. And if this centromere is like that, then it will turns to L shape. That means depending on the centromere, we can divide this chromosome structure in anaphase stage 4 shape, V, L, J, I. Then the telophase stage will start it. Let's see the telophase stage. Telophase stage is totally opposite to prophase. In prophase, what will happen? Dehydration occur, nuclear membrane and nucleolus start disappearing. In telophase stage, again hydration occur and nuclear membrane and nucleolus will start again appearing. Okay, in this here, again the nuclear membrane will come, nucleolus will start appearing, and due to hydration again the chromosome will turn into the chromatin structure. After that this occur in telophase stage. Let's see, at first let's see the anaphase. Let's read the anaphase stage. The centromere of the chromosome now splits so that two independent daughter chromosome, each with its own centromere, are formed. The chromatid gets separated from each other. At this stage, each chromatid is called daughter chromosome. In this movement, depending upon the location of centromere, chromosomes take different shapes such as V, L, J, I. Already I have discussed about that. Now come to telophase, that is this phase. Daughter chromosomes reach their opposite pole, spindle fiber disappears and the chromosomes lost their identity and reverting to a diffuse chromatin network like that. That means they again lost their structure because hydration occur here and again turns to chromatin. Finally, nuclear membrane reappears around the two daughter nuclei. Nucleolus also reappears. In this way, two daughter nuclei form in two pole and single end of karyokinesis. Look here, already two daughter nuclei already formed and already telophase stage has finished. After telophase, the cytokinesis will started. Look here, cytokinesis will started. Different cell material will start to appear. Different cell material will start to appear here. Different components of cell material, plasma membrane, cell plate will appear here, and finally look here, if you consider this picture, here the material will start to deposit and gradually it turns a specific form, and finally the plate, look here, finally it will cut like that and it will appear like that, and after that they will totally separated. After that they will totally separated and turns like that. And here also. They will cut the connection and become separated from one another. And after telophase, the karyokinesis finished, and after that the division of cytoplasm occur, and finally two different cell will produce. That means finally after cytokinesis the mitosis cell division will finish. Let's see the cytokinesis. At the end of telophase, cytokinesis starts. In plant cells, the equatorial region of the spindle apparatus gradually become white and touch to the cell wall, and the fiber disappear. If you consider here, in telophase already the spindle apparatus will disappear. Look here, the spindle apparatus, and gradually it will lost, and this will appear, this cell plate will appear. Here this cell plate will appear. At the equatorial region, phragmoplast of endoplasmic reticulum are deposited and they combine together to form a membrane called plasma membrane here at the middle. By deposition of other components of the plasma membrane, plasma lemma, cell plate is formed. Cell wall is developed by the deposition of hemicellulose and other component of the cell plate. By development of the cell wall, the mother cell divides into two daughter cells. That means after telophase karyokinesis will finish, then the cytokinesis started, and gradually here the cell plate and plasma membrane material will deposit, and finally it will cut the relation and separated from one another and turns to two different cells. In animal cell, a cleavage furrow appears on the surface of the dividing cell and encircles it at the middle layer of the spindle. The cleavage furrow dips and pinches the plasma membrane as through it are being tightened by an invisible rubber band and gradually draws the furrow inward. Finally, the infolding edges of the plasma membrane meet the fuse and completing cell division. In animal cell, that means in animal cell gradually it will shrink and finally it will cut like mitosis, which you will see, and then two different cells will produce. So dear students, here I have discussed the steps of mitosis. In mitosis, we know that nucleus and chromosome will divide only one time, and from a mother cell, two identical cell or two identical daughter cell will produce through mitosis, and it occur in two steps or two phases. At first karyokinesis occur, then cytokinesis occur. And in karyokinesis there are 5 steps: prophase, prometaphase, metaphase, anaphase and telophase. And after telophase the cytokinesis occur, new 2 cell produce, and finally the mitosis cell division completed. Now today here I will give you the SW from today's topic. Your SW number is 5 and you have to do 5 knowledge based question from this part. From today's topic, 5 knowledge based question from today's topic, and also you have to draw 5 phases of mitosis. You have to draw 5 phases of mitosis. That means you have to do 5 knowledge based question and you have to draw 5 phases of mitosis. Dear students, I hope you have understood today's topic. Please practice it in your home and keep practicing at home. Stay at home, stay safe. Thank you.